Hello, my name is Sophia Tang, and I will be talking about my project with Dr. Rania El-Sayed about the characterization and mapping of extracellular vesicles from oral tissue in periodontal health and disease. Periodontal disease is a chronic inflammatory disease caused by bacterial infection that leads to damage of the tooth-supporting structures. It is the leading cause of tooth loss in adults, affecting almost 50% of the adult population, and is linked to many other systemic diseases. To this day, the exact pathogenesis of the disease is unknown. Porphyromonas gingivalis, PG, has been recognized as a keystone pathogen of periodontal disease. A suggested mechanism of the disease with the role of helper T17 cells and dendritic cells has been mapped out by previous works in our lab. Dendritic cells are the resident immune cells in the gingival tissue linking the innate and adaptive systems. In this mechanism, the pathogen enters a dendritic cell and induces premature senescence or aging within that cell. Then the senescent dendritic cell releases cytokines, cell mediators, and exosomes as a part of its senescent-associated secretory phenotype, or the SASP, to the extracellular microenvironment. Released exosomes are potent in their capacity to invade other bystander dendritic cells, eventually reducing antigen presentation to the adaptive immune system. This paracrine amplification of the SASP creates an inflammatory microenvironment that will eventually induce bone loss. Exosomes are nanoparticles. They are membrane-bound vesicles secreted by various cell types and are essential for intercellular communication. They were first thought of as a method of waste disposal, but now discovered by Rothman, Sheckman, and Soidoff, are known to be important in cell-to-cell communications. Exosomes can carry a diverse array of biomolecules, and when exocytosed from parent cells, carry similar surface markers of the parent cell on their own membrane. They reflect the metabolic state of the parent cell inside the vesicle. The aim of this study was to characterize the exosomes released in gingival tissues in healthy and diseased states and identify their cellular origin, with a particular focus on age as a variable. Young and old mice were subjected to oral gavage either with PG, the periodontal pathogen, in the experimental group, or a sham infection in the control group over a 6-week period. Then the gingival tissue was harvested from the mice, and the exosomes were isolated from this tissue by enzymatic digestion and differential centrifugation. Exosomes from the experimental group were termed PD-exo, periodontal disease exosomes. Exosomes from the control group were termed con-exo, or control exosomes. Western blot analysis, nanoparticle tracking analysis, and electron microscopy were used to characterize the exosomes, and a semi-quantitative multiplex analysis was performed to reveal the cellular origin of the exosomes based on the expression of cell-specific markers on the exosomal membrane. This is the Western blot, NTA, and electron microscopy data. Western blot analysis revealed the presence of P. gingivalis antigens, MFA, and RGP in PD-exo, which are the exosomes isolated from the gum tissues of mice with periodontal disease. MFA is a bacterial fimbrial adhesion protein for proliferation and growth, and RGP is a proteolytic enzyme of P. gingivalis that destroys host tissues. CD81, CD9, and TSG101 were specific markers of exosomes, enforcing that these were in fact exosomes. The Western blot confirmed that the PD-exo showed the presence of periodontal disease in the exosomes. Nanoparticle tracking analysis, NTA, was used to verify the correct size of exosomes, between 30 and 150 nanometers in diameter. The NTA showed that a majority of the gingival exosomes were around 100 nanometers. This verified that the collected samples were exosomes. Nanogold plating for CD63, a protein on exosomes, was done on the PD-exo and observed with electron microscopy. The black dots at the end of the white arrow show the presence of CD63 and subsequently of exosomes. The multiplex analysis revealed that the expression of cell-specific markers on the exosomes varied with both the age of the subjects and the presence of periodontal infection. In comparing the exosomes of the old versus the young control group, the data from the flow cytometry showed the concentration of exosomes. In this bar graph, the exosomes of the old group, blue being old female and gray being old male, were compared to the exosomes of the young group, orange being young female and dark gray being young male. For the surface marker CD3, there was a higher concentration of exosomes in the old group than in the young group. CD3 is a surface marker for T cells. This showed that more exosomes were released from T cells in the old group than in the young group. As you can see, for the old mice, there was a relative increase of exosomes from leukocytes, including T cells, B cells, dendritic cells, and activated natural killer cells. This suggests an important role of lymphocytes and myeloid-derived exosomes in the aging of oral mucosa. Next, we can compare the exosomes from the control to the PD-infected animals. In this graph, orange is control exo and blue is PD exo. In the disease group, there were elevated levels in lymphocyte markers, dendritic cells, and platelets. This correlates with the important roles of T cells, B cells, dendritic cells, and platelets in periodontal disease. There was also a notable decrease in exosome expression of the epithelial cell marker CD326 and the stem cell marker prominent 1, which is known to have a role in periodontal regeneration. This suggests a role of these cells to be associated with the pathological progression of periodontal disease. To determine if this data can be extrapolated from mice to humans, this multiplex analysis was also performed on human exosomes, control and those with periodontal disease. In human PD exosomes, there were elevated levels of markers of T cells, B cells, natural killer cells, platelets, dendritic cells, lymphocytes, and monocytes. Data from human exosomes corroborated with our results from mice, where exosomes from leukocytes were significantly increased in gum tissue in periodontal disease. Results from this study were able to characterize the exosomes related to periodontal disease and show that leukocytes, like T cells and dendritic cells, are a major source of exosome release in the gingival tissue. These results provide valuable insights into the cellular origins of exosome release and the contexts of aging and periodontal disease. The study presents a potential role of these parental cells and the pathological processes associated with periodontal disease, and the findings also suggest promising avenues for therapeutic intervention aimed at preventing and treating periodontal disease, while enhancing our understanding of its underlying pathogenesis. Thank you so much to my mentor, Dr. El Sayed, and my lab members, Angelica, Dr. Chu, and Jasmine. Thank you.